Dear students, myself Satish Gowda, Assistant Professor and HOD, Department of Zoology, Government First Grade College of Arts, Science and Commerce, Shira. Today in this lecture, I am going to speak on a topic, brief account of Treponema pallidum. And this topic belongs to Tumkur University BSc Zoology 6th semester paper 7 Applied Zoology. A Treponema pallidum is a spirochete bacteria, one of the spirochete bacteria. Regarding this classification concerned, this is belongs to a bacterial domain, phylum Spirochetes, order Spirochetales, family Spirochetacea, genus Treponema, species Treponema pallidum. And this is the structure of Treponema pallidum as a helical structure and spirally coiled. Coming to the Treponema pallidum, a brief introduction, which is spirochete bacterium that is causing a dangerous disease, sexual disease, that is syphilis. Syphilis is a bacterial infection which is coming from the sexual contact, and it is transmitted only among the human beings, This Treponema pallidum is helical, as I told earlier, helical and coiled corkscrew shaped microorganism, and which is usually measures 6 to 15 micrometer long and 0.1 to 0.2 micrometer wider. And it is a motile spirochete. Motile means movement. It is a motile spirochete that is generally acquired by close sexual contact. They have an outer membrane which surrounds the periplasmic flagella. Multiplication of this bacteria is done by the asexual method of asexual reproduction, that is by binary fission. Coming to the pathogenesis. What is pathogenesis? Pathogenesis is the process by which the disease or disorder develops, is known as pathogenesis. Then how this Treponema pallidum causes the or develops the disease or causes the diseases? Treponemes are highly invasive pathogens which oftenly disseminate soon after inoculation, and evasion of host immune responses appear due to the unique structure of treponemal outer membrane, which consists of extremely low content of surface proteins as well as lipopolysaccharides, and they possess abundant lipoproteins which induce inflammatory process. These lipopolysaccharides, proteins and the lipoproteins are involving in the disease developing or invasive activity in the host body, and it affects the immune system of host body. And coming to the epidemiology. Epidemiology means the study of diseases. Here the Treponema pallidum epidemiology consists the study of epidemiological studies reveals the how this Treponema pallidum transmit and where it occurs first. So this epidemiological studies reveals that the sexually transmitted, this Treponema pallidum is sexually transmitted infections. It made sexually transmitted infections, that means STIs, and the role of commercial heterosexual sex networks in driving. So it is the STI transmission in the Middle MENA. Middle MENA means Middle East. Transmission in the Middle MENA means Middle East and North Africa regions. That is called as STA transmission in the MENA. MENA means Middle East and North Africa region, where this for the first time it transmit the sexual through the sexual contact, as the epidemiological studies says. And humans are the only source of treponemal infection. Treponemal infection. Venereal syphilis is the significant public health problem in many underdeveloped countries. So this venereal syphilis means the bacterial infection occurs at the genital regions, sexual genital regions, and the infections occurs at the rectum. This is known as venereal syphilis. So which is very significant, which causes very significant public health problem in many of the underdeveloped countries. Okay, then infectivity rates corresponds the most sexually active age groups. So this infective rates is corresponds the sexually active age groups. This is about epidemiology of Treponema pallidum, which causes venereal syphilis, and which was first occurred and transmitted by MENA in Middle East and North Africa region. Now coming to the diagnosis. Diagnosis includes the following methods. So here exudates and fluids from lesions are examined as a wet mount using dark field microscopy. Exudates means exudates, a group of cells present and the soaring fluid at the genital regions or lesions. From there, the wet mount using dark field microscopy examined by using the dark field microscopy. The identification of Treponema pallidum is based on the characteristic morphology and motility of spirochete. So to identify this Treponema pallidum, so it's need to characteristic features of morphology as well as motility rate of spirochete is very essential. And this method is suitable when lesions are moist, and the examination can be done immediately after specimen collection. So examination done by immediate collection of the specimen from the infected person. Then finding of treponemes within exudative lesions and positive serology aids the diagnosis. So this finding of treponemes within the exudative lesions means the leakage of fluids from the lesions, that is called exudative lesions, and where it is positive serology is also helps to diagnose the Treponema pallidum disease, that is syphilis. This is about diagnosis. And coming to the transmission, means the transfer the pathogen Treponema pallidum from one person to another person. How we to transfer? It is transferred by the sexual intercourse. Transmission occurs during the sexual intercourse. So during sexual intercourse, it is very easily transferred from person to person, and it also transfer by blood transfusion during emergency cases. During the blood transfusion, it may transfer from person to person. It can also be introduced into an open wound in the skin. ಅಲ್ಲಾದರೂ ಓಪನ್ ವೂಂಡ್ ಆಗಿದ್ರೆ ಅದರಿಂದಲೂ ಕೂಡ ಅದನ್ನು ಟಚ್ ಮಾಡೋದಾಗಲಿ ಇದು ಮಾಡೋದಾಗಲಿ ಮಾಡಿದ್ರೆ ಸೊ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗೋ ಚಾನ್ಸಸ್ ಭಾಳ ಇದೆ. In the region of venereal inflammations or the mouth region or rectum region, if the open wounds are present, during that the person's contact with that open wound, it may transfer from person to person. And how the controlling measures helps to control the Treponema pallidum or the disease syphilis, venereal syphilis. So control of venereal treponematosis is based upon active surveillance and treatment of contacts. So active surveillance and treatment of contacts helps to control the disease venereal syphilis. Means we can control this disease by controlling the venereal inflammations or sexual contacts, or by testing the blood during blood transfusion, etc. And after infected, the treatment should be there. Treatment is given by using antibiotics like penicillin. Penicillin treatment eradicates all stages. This penicillin injection is there, no? Penicillin eradicates all the stages, including congenital infection. So this is about the brief account of Treponema pallidum. Treponema pallidum is a bacteria, spirochete bacteria. This bacteria causes venereal syphilis. So this syphilis is a disease where it caused or occurs during the sexual contact with the unknown person, and it transfer from parents to parent by the blood transfusion as well as sexual intercourse. And this Treponema pallidum is very dangerous bacteria which cause sexual disease, that is called as syphilis. Hence we all need to aware of this dangerous bacteria, that is Treponema pallidum. I hope all you are understood well about Treponema pallidum bacteria, and I will end up this session here, and I will meet you in the next session with different topic. ಅಲ್ಲಿವರೆಗೂ ಆರೋಗ್ಯವಾಗಿರಿ ಮತ್ತು ಸೇಫಾಗಿ ಮನೆಯಲ್ಲೇ ಇರಿ. ಥ್ಯಾಂಕ್ ಯು ವೆರಿ ಮಚ್.